Hello there. Welcome back to Dr. K. Prem Primer Lecture Series presenting by Dr. K. Prem. That's me. Today we will discuss about DNase I, right? So I want to ask you one question. Whatever we are eating every day, like they contain carbohydrates, proteins, lipids, and DNA. It may be the vegetables or it may be the chicken or mutton. They consist of these four macromolecules. So proteins are degraded by proteases into the amino acids. And the lipids are again into the lipases into the fatty acids and glyceroldehydes and so on. Then coming to the carbohydrates, amylases degrade them into uh, oligosaccharides or disaccharides or monosaccharides like glucose molecules. But what about DNA? So when we are eating vegetables or chicken or a mutton or fish, they, they consist of all the four macromolecules, including DNA and RNA also. So DNA is degraded by a digestive enzyme called as DNase I. So whatever DNA we take, that's a foreign DNA from vegetables or other source of food or grains, that DNA is degraded in the intestine by DNase I. This is the most prevalent enzyme in the mammalians as well as the mammalians. So DNase is secreted by pancreas. That pancreas secretes the DNase, and that secreted DNase one breakdowns the DNA molecules, whether they are single strand or double strand. So this is the one of the DNA modifying enzymes frequently used in RDN technology and molecular biology. So in today's lecture, we will talk about the mechanism of DNase I, activity mechanism, then followed by what is the properties of DNase I. Then we will see the what are the applications of DNase I in RDNA technology. As you all aware that polynucleotides are connected by a bond called as phosphodiester bond. You see, this is the adenine which is having connected to the ribose and phosphate. This is a nucleotide. So it is connected to the other nucleotide, like thymine, ribose and other through the phosphodiester bond. Ester bond is COP. There are two COPs, hence it is called as phosphodiester bond. So polynucleotides are connected by phosphodiester bonds, right? This is a phosphodiester bond. So here DNase to break down the polynucleotides into nucleotides, they have to break down the phosphodiester bond, right? So DNase I, this is a DNase I, is having a calcium and magnesium are the cofactors. So calcium is absolutely required for structure and function. Magnesium enhance the activity of DNase I, right? So DNase breakdowns the one of the phosphodiester bond, like so you see two monoester bonds are there, right? So this DNase break downs the one of the COP bond, like one of the COP and COP bond, right? That's a ester bond. So this phosphodiester bond. So here the DNase break downs the one of the COP bond and separates the nucleotides, right? You see, same thing here, right? So here you can see adenine and thymine, which are mononucleotides, are released after digestion with the DNase I, right? So when they released, so the mononucleotides tend to consist of 5' phosphates, 5' monophosphate, and 3' hydroxyl group. Whenever polynucleotide is digested with the DNase I, and it releases the mononucleotides or trinucleotides or tetranucleotides or oligonucleotides. But their pi prime is having one phosphate, and it is also 3 prime is having the OH molecules. This is how the DNA digests the polynucleotide into the nucleotides with monophosphate, monophosphate, monophosphate at the pi prime and 3 prime OH, right? Okay. Now we will see DNase I acts on single strand DNA molecules as well as it can act on the double strand DNA molecules, right? It is a endonuclease, so it can cut within the DNA and non-specific cleavage or non-specific breakdown of phosphodiester bond. So you see, all over the DNA it will cut, right? So DNase I acts on single strands and double strands randomly, or non-specific cleavage happens with the DNase I, right? So once it is digests the single strand or double strand DNA, then it releases into the monophosphates or diphosphates, mononucleotides or dinucleotides, trinucleotides or tetranucleotides, or sometimes oligonucleotides also. DNase also have one more activity is called as oligonucleotide nuclease. So whenever it releases the oligonucleotide, that is further degraded into the mononucleotides or dinucleotides or tetranucleotides, right? So if you give a DNA and if you digest with the DNase I, and ultimately at the end of the reaction, you will have a mononucleotides or dinucleotides or tetranucleotides, right? You see? After digestion, you can that releases the mononucleotides, di or trinucleotides, sometimes oligonucleotides also. Oligonucleotides are further degraded into the mono, tri, or tetranucleotides. But all the mononucleotides or tetranucleotides or tri or dinucleotides have a monophosphate at 5 prime and 3 prime OH. So whenever DNase I digests the DNA, it releases the nucleotides with the 5 prime monophosphate and 3 prime OH. That's a hallmark of the products, right? And I hope fully you understood. And now we will talk about the few features of DNase I. And you see, is a nuclease. Basically, it is a nuclease. It's also called as phosphodiesterase that cleaves the one of the two bridging PO bonds, CPO bonds, in a nucleic acid polymer. And DNase I is an endonuclease, make the nicks along or inside the DNA, right? It can cleave non-specifically, as I told you that it cleaves the DNA non-specifically or randomly, both single strand or double strand DNA, and they break down by breaking the phosphodiester bond and releases tri oligonucleotides with 5 prime monophosphate and 3 prime hydroxyl ends. So the products have 5 prime monophosphate and 3 prime OH hydroxyl groups. Yeah. Not only this one, DNA not only acts on DNA in extracellular space, but also within the cell. Whenever cell is undergoing the apoptosis, so that time DNase I degrades or fragments the chromatin molecule, chromatin. So it plays whenever cell is undergoing the apoptosis, that's nothing but programmed cell death, where cell undergoes the shrinkage and then followed by the condensation. So cell is fragmented in that particular phase. So even DNA also fragmented into small portion. So where the DNase I plays a role by fragmenting the DNA. In DNA and RNA hybrid also, it can digest the DNA, right? So physiologically, DNase I plays a role in digestion of DNA in extracellular space that we consume that through the, right. And second thing is that it plays an important role in the in the apoptosis where it degrades the DNA, right. And also it is involved in the base accession repair also, right. And you see, DNase I is the most prevalent in mammals and also found in bacteria, but not in lower eukaryotes or plants. So it is present in the mammals and bacteria, but not in the lower eukaryotes and plants. Yeah, here I told you that in the beginning of it is required the two metals as a cofactor. One is the calcium, other one is the magnesium. So calcium cations protects DNase I from proteolytic degradation, and it is two sulfide bridges from reduction. So absolutely calcium is very, very essential for DNase I because it is protecting the DNA from proteolytic degradation, and it is also protecting its two sulfide bridges. Hence, you cannot imagine the DNase activity in the absence of calcium. Whenever DNA is treated with the EDTA or EGTA, which are metal chelators, they will chelate the calcium from the enzyme. Then the enzyme becomes inactive, right? So DNase is absolutely depends on calcium for its structure and activity. And in addition to the magnesium, in addition to the calcium, it also requires the magnesium. So hence they are called two metal nucleases. DNase is a two metal nuclease. Then you see, as I told you that pancreas are the rich source of DNase I. And pancreatic DNase is among the first nuclease discovered and characterized. And it's a glycoprotein. It's basically DNase is a glycoprotein with a molecular weight of 31 kDa. And it is in bovine, it is having 4 isozymes A, B, C, D. And sometimes, it prefer to digest the DNA around the pyrimidines, right? So DNase is inactivated by incubating at 65 to 75 degrees centigrade for 5 minutes. If you incubate that, then that enzyme is inactivated. So it is DNase is measured in the Kunz unit. Kunz unit means 1 unit is the amount of enzyme required to cause an increase of 0.001 at 260 nanometers per minute per ml at 25 degrees centigrade in sodium acetate, 0.1 molar of sodium acetate, pH 5, due to degradation of polymerized DNA. So Kunz unit means the amount of enzyme is required to increase the absorbance 0.001, right, per at 260 nanometers. So when you provide the polymeric or polymerized DNA, so DNase breakdowns the polymerized DNA into mono or di or tetranucleotides. When DNA is degraded into the nucleotides, its absorbance is increased. So increase in the absorbance is recorded at the 260 nanometers, right? So the amount of enzyme which is required to increase the absorbance of 0.001 minute at 260 nanometers per minute and ml at 25 degrees centigrade, right? That enzyme is that amount of enzyme is called as one Kunz unit. And you see degradation assays. One unit is defined as the amount of enzyme required to completely degrade the 1 microgram of plasmid DNA in the 10 minutes at 37 degrees centigrade in 10 millimolar TrisHCl, pH 7.5, and 50 millimolar MgCl2, and 13 millimolar of calcium chloride. So you can see here in the degradation assay, magnesium chloride is provided. You see, the magnesium chloride is provided and calcium chloride also provided. Why? Because the enzyme depends on the calcium presence as well as the magnesium presence for reactivity. Hence, in the buffer, a buffer you are taking for enzyme activity, then it is having both metal ions, calcium and magnesium, right? This is a degradation assay. Now we will see the applications of DNase in rDNA technology. The first one is used in RNA preparation to digest the contaminating genomic DNA. Whenever we are isolating the RNA from different tissues or cells, so DNA comes as a contamination. So when a DNA is present in the RNA, so then that is not suitable for many applications, downstream applications, such as if you want to do the real-time PCR, real-time ECR. So for what you want to know, you want to characterize the gene expression of particular gene. So this genomic DNA will have an impact on that, right? So if you want to do northern blot also, you don't want genomic DNA must not be there. So in that case, whenever you are preparing a RNA, total RNA or mRNA, then what you do is, once you isolate the RNA, then on column DNA degradation assays are available. So that on column DNA is digestion assay will perform, then the preparation of RNA is free from the genomic DNA. Genomic DNA. The second important thing is that used in DNA footprinting analysis. So you all know that DNA, DNA where promoter region is occupied or binds by the transcription factor, or you want to know the transcription factor binding site in the promoter region, right? If that region is protected because that region is occupied or by the transcription factor. So in identification of DNA protein interaction, to know the DNA protein interactions, so whenever DNA is occupied by the certain proteins, then if you that region is subjected to DNA, then wherever there is a protein, that region is protected and remaining region is degraded. So it indicates that the protected region is having a transcription binding sites. So this is what is the used in DNA footprinting analysis or DNA protein interactions, especially transcription factors or other enzymes which would bind to the DNA, right? And third one is DNase is used in the preparation of probes through the Nick translation. Nick translation, where the DNA is partially digested with the DNase I. Partially digested means it will only form a Nick or less enzyme or small time. If you keep the DNA digestion assay only for small time or less enzyme, then it forms a Nicks in a Nicks. So that Nicks, from that Nick, the probe is prepared with the clean-out enzyme or DNA polymerase. So used in Nick translation for preparation of probes or partial digestion of template makes it Nicks. And the last one is DNA sequencing to make a fragments. You have two big fragments. So those two big fragments cannot be sequenced. You need a smaller fragments. So if you do digestion of DNA of your interest with the DNase I, you get a partial, you do the partial digestion, you get a smaller fragments, like 2 kb to 3 kb. Such DNA fragments can be used to for DNA sequencing through the Sanger's method. So this is all about the DNase and DNase mechanism and DNase I properties and the applications in RDNA
technology. If at all you like my video, try to subscribe for regular updates. Thank you all for listening, and see you soon with one more video. Until then, bye bye.